Antibiotic sensitivity testing. To determine the antibiotic sensitivity of bacteria, the following method is used. Soak a sterile swab in a diluted bacterial culture and press it against the inner wall of the tube to remove excess fluid. Carefully swab the entire surface of the Mueller-Hinton agar plate, ensuring that the swab overlaps each time to evenly distribute the inoculum. Turn the plate 120 degrees and swab again. Repeat a third time. Place the used swab in a swan bag. Then, using the antibiotic disc dispenser, stamp the discs over the swabbed surface. Incubate at 37 degrees overnight. After overnight incubation, examine your Mueller-Hinton plate. If zones appear around the discs, then the organism is sensitive to the antibiotic. However, if no zones appear around the discs, then the organism is resistant to the antibiotic. Sensitivity patterns can also be measured and recorded as either sensitive or resistant by measuring the inhibition zone size and comparing the results against a reference table of antibiotic zone sizes. If the measured diameter of the zone is larger than the inhibition zone diameter from the reference table, it is then recorded as sensitive.
The biochemical tests used for first stage identification are the catalase test, the oxidase test, and the coagulase test. The catalase test. Hydrogen peroxide is the reagent used in this test. Catalase is an enzyme possessed by certain bacteria which changes hydrogen peroxide to water and oxygen. Step 1. Adding hydrogen peroxide to the slide. Firstly, an inoculating loop is sterilised in the Bunsen flame. It is then used to pick up a drop of hydrogen peroxide. The hydrogen peroxide is placed onto a clean glass slide, and the loop is sterilised again. Step 2. Adding a bacterial colony to the slide. A sterile inoculating loop is used to pick up a bacterial colony from an agar plate. This is added to the drop of hydrogen peroxide on the slide. The black tile under the slide helps to see whether the result is positive or negative. Positive result. Bubbles of oxygen gas forming in a few seconds indicate a positive result. Negative result. If no bubbles form after the colony has been added, the result is negative. Here we compare positive with negative. Oxygen bubbles are clearly visible on the slide to the left. Oxidase test. Commercially available oxidase paper is impregnated with the oxidase reagent. Step 1. Streaking a bacterial colony onto oxidase paper. Using a sterile inoculating loop, an isolated bacterial colony is removed from an agar plate. The colony is then streaked firmly across the surface of the oxidase paper. The loop is then sterilised again. Result: negative versus positive. If the organism is oxidase negative, as in the window on the top left, no colour change occurs. However, if an oxidase positive organism is tested, a blue colour appears within 30 seconds. This is seen here in the top right-hand window. Coagulase test. Rabbit plasma is the reagent used to test for the coagulase enzyme. Step 1. Adding saline to the slide. A sterile inoculating loop is used to obtain a drop of sterile saline from a jar. This is then placed on a clean glass slide and the loop is re-sterilised in the blue flame. Step 2. Adding a bacterial colony to the slide. A sterile inoculating loop is used to remove an isolated colony from an agar plate. It is mixed with a drop of saline on the slide to form a milky white suspension. Step 3. Adding rabbit plasma to the slide. Using a sterile inoculating loop, a drop of rabbit plasma is removed from a bottle. The drop of rabbit plasma is placed on the slide next to the suspension. The two drops are mixed together a little so that they overlap slightly. Step 4. Rocking the slide back and forth. The slide is then carefully picked up and gently rocked back and forth. If tiny white granules are seen to form, then the organism is coagulase positive. Here, a large white clump is visible. Thus, this organism is coagulase positive. However, the result of this test is often hard to visually assess. No clumps are seen in the suspension on the left. Thus, this organism is coagulase negative. Many tiny white clumps are visible in the suspension on the right. Thus, this organism is coagulase positive. Biochemical tests. Second stage identification. The biochemical tests used for second stage identification include the following: the IMVIC tests, the TSI lysine decarboxylase tests, Loeffler's serum slope, and the motility test. IMVIC tests. These are used to differentiate between members of the Enterobacteriaceae. They consist of the indole test, the methyl red test, the Vogt-Prosker test, and the citrate test. The indole test. An uninoculated indole test appears straw-coloured. A negative test appears straw-coloured with a straw-coloured ring at the surface. A positive indole test can be recognised by the red ring at the surface. The methyl red test. An uninoculated methyl red test appears clear and colourless. A negative result is straw-coloured, while a positive methyl red test appears pink. The Vogt-Prosker test. An uninoculated Vogt-Prosker test appears clear and colourless. A negative result is indicated by a straw colour, while a positive result is pink in colour. The citrate test. An uninoculated citrate slope is green. A negative citrate slope is also green in colour, whereas a blue citrate slope indicates a positive result. Here are the IMVIC test results for E. coli: indole positive, methyl red positive, Vogt-Prosker negative, and citrate negative. Here are the IMVIC results for Klebsiella: indole negative, methyl red negative, Vogt-Prosker positive, and citrate positive. The TSI and lysine decarboxylase tests. These are done to identify Salmonella species. This procedure requires uninoculated lysine broth and a tube of uninoculated TSI agar. The colonies to be tested are suspect Salmonella colonies growing on XLD agar. Step 1. Inoculation of the TSI slant. A sterilised straight wire is used to remove a bacterial colony from a plate of XLD agar. The wire is then used to inoculate the TSI agar. This is called stab inoculation, as the wire is inserted directly into the agar. It must be dragged up the slope as it's being removed and wiggled from side to side across the agar surface. Step 2. Inoculation of the lysine broth. The The same wire, which still carries residual bacteria on it, is then used to inoculate the lysine broth. This is done simply by dipping the wire into the broth and stirring it around. Note that the wire is not held in the Bunsen flame between the two inoculations. The two tests are then incubated for 16 to 18 hours at 37 degrees Celsius. The lid of the TSI agar is left loose during incubation. Possible results. Uninoculated lysine broth is clear and dull purple in colour. A positive lysine decarboxylase result is bright purple and turbid, while a negative result is straw-coloured. The TSI result for Salmonella appears as a red alkaline slant and a yellow acid butt, with black hydrogen sulfide superimposed on both colour reactions. The TSI tube for E. coli appears as follows: a yellow acid slant and butt, and no black hydrogen sulfide. Loeffler's serum slope. An uninoculated Loeffler's serum slope is smooth and non-pitted. A negative result also appears smooth and non-pitted. A positive result has a dip or pit in the smooth surface of the agar slant. This pit reveals the presence of protease-producing bacteria, as enzymes produced by the bacteria digest the proteins present in the serum slant. Motility test. This detects whether bacteria are motile or non-motile. A specific kind of agar, known as motility agar, is needed for this procedure. It is semi-solid and contains tetrazolium salts. Step 1. Picking up a bacterial colony with a straight wire. Firstly, a straight wire is sterilized. Then it is used to pick up an isolated colony from an agar plate. Step 2. Stab inoculating the motility agar. The motility agar is then stab inoculated with a straight wire. Wherever the bacteria grow, they produce an insoluble red pigment from the tetrazolium salts. Thus, if bacteria are motile and spread throughout the tube, this will appear as a colour change. Possible results. Uninoculated motility agar appears pale and straw-coloured. Non-motile bacteria appear as a dark reddish line along the initial area of inoculation, as they can't spread out. Motile bacteria turn the whole tube a cloudy red colour as they move throughout the agar.
Edvotech instructional videos. The enzyme-linked immunosorbent assay, or ELISA. For this experiment, you will need a microtiter plate, phosphate buffered saline, antibody and antigen samples, a color-changing substrate solution, a 37-degree incubator, an adjustable micropipette and tips, and transfer pipettes. Step 1. Label the microtiter plate and transfer pipettes as noted in the product literature. Step 2. Add 100 microliters of the antigen solution to all of the wells. Step 3. Incubate for 5 minutes at room temperature. At this time, the antigens will non-specifically adhere to the plastic through hydrophobic and electrostatic interactions. Step 4. Remove all the liquid from the wells using a transfer pipette. Step 5. Wash the wells by adding approximately 8 drops of PBS buffer to each well. Remove all the PBS from each of the wells, taking care to prevent the buffer from spilling over into adjacent wells. Traditionally, following this step, the wells are blocked with a protein-containing buffer to prevent non-specific interactions between the antibody and the plastic wells. We have optimized this experiment to eliminate this step. Step 6. Add reagents as outlined in the product literature. Remember to use a clean micropipette tip for each reagent. Step 7. Incubate the plate for 15 minutes at 37 degrees Celsius. Step 8. Remove the liquid from the wells using the appropriately labeled transfer pipette. Step 9. Wash each well with fresh PBS. Remove the liquid using the transfer pipette designated for each sample. Step 10. Add 100 microliters of the secondary antibody to each of the wells. Step 11. Incubate for 15 minutes at 37 degrees Celsius. Step 12. While the samples are incubating, prepare the detection substrate as specified in the product literature. Step 13. Remove the secondary antibody solution from the wells using the transfer pipette designated for each sample. Wash each well once with fresh PBS buffer. Remove the liquid using the transfer pipette designated for each sample. Step 14. Add 100 microliters of the substrate solution to each well. Step 15. Incubate at 37 degrees Celsius for 5 minutes. Step 16. Examine your results. Differences between negative and positive samples will be obvious, with most positive samples turning brown in color. If color is not fully developed after 5 minutes, incubate at 37 degrees Celsius for a longer period of time.
Hello everyone, welcome back to Synthetic Biology 1. Today it's time for some colony PCR. Colony PCR is a very useful technique to amplify any type of specific sequence on either the DNA of the genome of a bacteria or a plasmid. Yesterday I transformed my bacteria with a plasmid, and now I want to use colony PCR to check if the plasmid is actually there. Let's get my Petri dishes from the incubator. Oops. So here I have 2 plates. One plate with colonies that should have the plasmid that I transformed. And this plate has colonies from a bacterial strain, the same strain as this one, but without the plasmid. And I'm going to use this as a negative control. So before I can start to prepare the PCR reaction mix, I first have to release the DNA from these cells, because otherwise they're not accessible for the polymerases that need to amplify the DNA. I'm going to mark 3 colonies on this plate with the bacteria that carry my plasmid to know which colonies I actually checked. So let's pick this one. This is number 2. And here I have number 3. And I'm going to use this negative control colony. Alright. So I also labeled some PCR tubes with the same numbers to know exactly which colony went into which PCR tube. The idea is to take a small volume of water So I'm taking 10 microliters here. And here I have some wooden sticks that I can use to pick each colony. And just dip it into the liquid, mix a little bit with the stick, and then you're good. So I'm doing this for 2 other ones. And finally, the negative control. So that's step 1. Now we need to go to the PCR machine to heat the different tubes. Put them inside the machine, and I set the temperature at 98 degrees. 10 minutes should be good. I press start. Okay, so this is done. Now I can start with the preparation of the PCR mix. So in this protocol I'm going to use a Taq polymerase. This Taq polymerase lacks proofreading activity, meaning that it doesn't check if it made a mistake during the DNA polymerization. But for this case I don't really care, because I'm only going to visualize my DNA fragments on gel, and I'm not going to use the DNA later on in other experiments, for example to construct a plasmid. So if you're interested in having an amplified sequence that is very correct, I would recommend you to use different polymerase, for example a high fidelity polymerase, not the Taq. But the advantage is that for the Taq polymerase we have already a pre-mixed cocktail that contains all the components, all the reagents that we need to do this PCR mix using Taq. And that will save us some pipetting steps. As you saw before, I have 4 different tubes, so I want to do 4 individual PCR reactions. And instead of making every reaction separately, I first will prepare a master mix. This master mix will contain all the components for the reaction, and later on I will divide the master mix into 4 tubes for each individual PCR reaction. So what we need is, of course, our DreamTech mix, some water, a forward primer and a reverse primer. So I already made the calculation to know how much of each component I need to add in order to be sufficient for 4 reactions. And so I will add 26 microliters of water, ten microliters of my reverse primer, ten microliters of my forward primer, and finally 50 microliters of the DreamTech master mix. Okay. So this is good. Then I'll take some PCR tubes. So I labeled my my tubes with 1, 2, 3 and with a minus for the negative control, and I will now add to each tube 24 microliters of master mix. Okay, so I think my bacterial cells are ready. Yes, they are. So I take them out and I will add 1 microliter of every heated bacterial solution to every tube that contains the PCR reaction mix. So number 1 goes in number 1. Number 2 goes in number 2. Number 3, 3, and finally the negative control in the 4th tube. So that's it. Oh wait. Yes, our tubes are ready. The cells are inside the PCR reaction mix, and the only thing we need to do now is take them to the PCR machine, place them inside, and I will set the temperatures as following. So I'm going to use in the first step 95 degrees for 3 minutes. Then in the second step, to open DNA, 95 degrees for 30 seconds. Then in the third step, I'm going to use 55 degrees for 30 seconds to make the primers anneal to the DNA. In the fourth step, I'm going to use 72 degrees for 1 minute, because it takes more or less 1 minute to amplify a DNA sequence of 1000 base pairs. And in my case, the DNA fragment that I expect to see is indeed around this number of base pairs. Then step 2 until 4 are repeated for 30 times. And finally, we have a final extension step at 72 degrees for 5 minutes. And then we'll end the reaction by cooling it down. My volume is 25 microliters, as you can see here. So I think we're good to go. I can press run. Okay, and there we go. So we're about 3 hours later and our PCR is done. I'm going to take them out of the machine. So the tubes are done, and the next step for our colony PCR would be to visualize them on an agarose gel. So I would load the samples into the gel and check the size of the fragments to see if the correct one is present. But of course you could also use it to, in other situations, you could also use these reactions in other downstream applications. Maybe you just want to purify the PCR reaction directly using a cleanup kit, or maybe you want to sequence the DNA that you amplified. So this is it for now. I hope you learned a lot, and I hope to see you next time.